Hello and welcome to the Pencil Tutors channel. I am Nandigga and today I will be giving a biology lesson on the meristematic tissues and growth. Meristematic tissues are basically undifferentiated living tissues in plants which are responsible for growth. There are two types of meristematic tissues, which are the apical meristems and the lateral meristems. The apical meristem tissues are located in the tips of the plant shoots and roots, whereas lateral meristems consist of cork cambium and vascular cambium. The tips of shoots and roots can be divided into three zones of cell growth, which is the zone of cell division, the zone of cell elongation, and the zone of cell differentiation. The positions of each zone is different for the shoots and for the roots. For the shoot, the zone of cell division is on the top of the shoot, whereas for the roots, the zone of cell division is at the end of the bottom. For zone of cell elongation, it is both in the middle of the shoot and in the middle of the root. For the zone of cell differentiation, in the shoots, it is at the bottommost part, but in the roots, the zone of cell differentiation is at the uppermost part. Now, let's take a closer look at each of the zone of cell growth. First of all, we have the zone of cell division. The zone of cell division usually takes place at the apical meristems that consist of actively dividing meristem cells that undergo mitosis. This is to increase the number of cells and cause an elongation of the plant stem. When new cells are forming, the cells formed previously are pushed to the zone of cell elongation. Next, we have the zone of cell elongation. This zone consists of cells that are increasing in size. The increase in size happens through water diffusion by osmosis and the absorption of nutrients into the cells and stored in vacuoles, as you can see in this diagram. The small vacuoles will fuse together to form larger vacuoles. This process is called vacuolation. The diffused water exerts pressure against the cell wall. This then pushes, elongates, and widens the cell. Last but not least, we have the zone of cell differentiation. This zone consists of differentiating cells that differentiate once they have reached their maximum size. The cells differentiate to form permanent tissues such as the epidermis, the cortex, the xylem, and the phloem. Cells will change their shape and structures to become specialized cells with specific functions. For example, the epidermal cell in leaves will differentiate and form guard cells. This is to control the opening and closing of stomata. Other than that, the roots will differentiate to form root hair cells. Next, we are going to look at the types of growth in plants. There are two types of growth in plants, which is the primary growth and the secondary growth. First, let's take a look at primary growth. Primary growth occurs after germination and takes place in all plants to elongate their stems and roots. This growth takes place at the apical meristem of shoot tips and root tips. The primary growth starts when meristem cells in the zone of cell division of apical meristems are actively dividing. This is then followed by elongation and differentiation. The leaf primordia and shoot primordia will grow and form new leaves and shoots. This will increase the plant's height. The root tips, on the other hand, have root caps that become exhausted when they penetrate the soil. This causes the cell of the root cap to be replaced by meristem cells. Now, let's take a look at secondary growth. Secondary growth occurs mainly in eudicots and small number of monocots, like shrubs. Secondary growth is to increase the circumference and diameter of a plant stem and root. For non-woody plants such as herbaceous plants, secondary growth does not happen. As you can see in this diagram, Secondary growth results from the division of lateral meristem cells that are located in the stem and root. The lateral meristems consist of vascular cambium and cork cambium. Vascular cambium is located between the phloem and xylem tissues in the vascular bundle, whereas the cork cambium is located under the epidermal layer of the plant. Now, we are going to take a closer look at secondary growth within the stem. This diagram shows the growth of plants for 3 years. As you can see, in the first year, the plant is non-woody, but as it goes on to the 3rd year, it develops a woody layer. This is known as secondary growth. I am going to teach you 4 steps of the secondary growth at stems. First of all, the secondary growth starts with the vascular cambium that starts to divide actively through mitosis. Then, the cells in the cambium ring will divide inward and form new xylem and outward to form new phloem. The new xylem tissue will become a secondary xylem, whereas the new phloem tissue will become a secondary phloem. After that, when the secondary xylem layer is compressed, there is an addition to the circumference of the stem. The stem will stretch and crack, as seen in the diagram before this. The cork cambium actively divides, forming cork cells on the outer side and cortex on the inner side, as you can see in this diagram. Lastly, when undergoing secondary growth, the primary xylem is pushed towards the pith, whereas the primary phloem is pushed towards the epidermis of the plant. The primary xylem tissues are compressed to form a strong woody layer at the outer surface. This is because the xylem walls are thickened with lignin, and this will provide support to the plant. Now, let's take a look at secondary growth within a root. First of all, the vascular cambium of the cell divides actively and combines to form a complete ring, as you can see in this diagram. The cells in the cambium ring divide inwards to form a secondary xylem and will divide outwards to form a secondary phloem. Due to the vascular cambium activity, the root will become thicker, as you can see in the diagram above. The cork cambium that is located under the epidermis will divide actively to form cork cells, whereas the cork cells provide protection to the root tissues. Last but not least, we have the secondary growth of monocots. Not all monocots undergo secondary growth, but some do. For example, the Dracaena species, the aloe species, and the agave species. We have finally reached the end of our lesson. But before we end, let's answer a few questions. First question, diagram 7 shows the longitudinal section of a root tip. This is a root tip. Which zone undergoes mitosis? First of all, let's label each and every one of the zones. The zone on the top is the zone of cell differentiation. The zone in the middle is the zone of cell elongation, and the zone at the bottom is the zone of cell division. So, the zone where cells undergo mitosis would be, yes, the zone of cell division. Therefore, the answer is C. Next question. Diagram 26 shows the cross-section of a dicotyledonous stem after secondary growth. Note that it is after secondary growth. What are structures P and Q? Can you remember what are structures P and Q? I'll give you a few seconds to think about your answer before I reveal it. Have you thought about it? If you answered A, then you are correct. The answer is P is the secondary phloem, while Q is the primary xylem. That would be all from me today. So thank you for watching the video and be sure to like this video and subscribe to our channel. Don't forget we post videos every Monday, Wednesday and Friday covering physics, mathematics and biology. Bye bye!